Okay, so the second page of this document starts with polymers with a blank. Polymers are created by a condensation reaction. So every molecule we've talked about, including water, are involved in a condensation reaction. We've already seen how lipids are formed, or at least a triglyceride is formed. Let's take a look at how carbohydrates are formed. And again, it's all by condensation reaction, spitting out of water. So all of these molecules are not complete. Both of these molecules are not complete. They all have OHs hanging off the end, but we're going to focus only on these OH groups because these are the ones that are going to interact, causing these molecules to come together. So we have a glucose and a fructose, or fructose, whatever you want to call it. They're going to come together. Condensation reaction means it's going to spit out of water, so take your pick. H and OH groups, H from one and OH from another, are going to come out. There's our water. And so now this carbon, which was bonded to this OH group needs something to bond to, and there it is, conveniently enough. So we just formed a disaccharide. Here's two monosaccharides to form a disaccharide. Two monosaccharides are glucose and fructose. The disaccharide is sucrose, table sugar. And again, what comes out of that is a water molecule. This was sticking in there, H from one and OH from another. Comes out to form condensation reaction to form this disaccharide. To undo this, we'll see this in a little bit, to undo this, we just add the water back in. Okay, we add the H to the O. We add the H to the O. Okay, just adding the water. And the OH comes in and replaces that bond. Essentially breaks it apart into its parts again. Okay, so back to our worksheet. The monomers of carbohydrates, hopefully easy to remember because of the beginning here. Mono, one, and remember Greek for sugar right there. So their monosaccharides are the monomers of carbohydrates. There's some examples for you. And moving on, nucleic acids. Monomers are nucleotides. Oops, misspelled. Nucleotides. And the monomers of proteins are amino acids. Example that I give in class is tryptophan, and if you watch the other video, there's a pirate's favorite amino acid. And then I inserted a glucose, just a simple sugar. There's our example. No need to label this guy. Okay, it's just a ring shape, has a bunch of C's, H's, and O's. Don't forget, each one of these king points here is a C and an H. Okay, that H here with a C is fulfilling that fourth bond for this carbon. So this carbon up here is attached to this oxygen. That's one. Attached to this carbon. That's one. Attached down here to this carbon. That's one. So that's three. Also attached to an H. So each one of these king points here are C's and an H, but again the H's aren't as important. There's our nucleotide, which has three parts that are not labeled on here, so this would be a good time to pause it. Or after. Your labels here, phosphate group in the circle. If you just want to put a P, later on you can write it, or off to the side. If you just want to put a P inside here, if you can smash phosphate off to the side, that's good too. Phosphate, sugar, right here. If you just want to draw an arrow from down here, that would work too. Or just label it with an S and then later draw an arrow. And here are here is your nitrogenous base, just made up a bunch of N's. Where there were C's and H's, there are N's and H's. Okay, but important to see the nitrogens there because that's what gives it away as a nitrogenous base. Without those nitrogens there, it kind of looks like a sugar carbohydrate. And these might not be in the right spot. I just threw them in there just so you can see that it's made of nitrogens. Alright, then amino acid. Again, made up of that R group, which is varied for each amino acid of the 20 amino acids that life uses. Each amino acid has a different R group, but the similarities, COOH group right here, carboxyl group, and the NH2 or amine group, where we get the name amino acid. So smashed in there. There's our amino acid. And guys, you should recognize these structures for the test. You should recognize them like this. You know, maybe CH, CH, CH here, so you would, if you needed to, count them. You know, when we get down to this guy, if you needed to count these numbers of C's, H's, and O's, you could do it. Okay, this is 1 part carbon, 1 part water, H2O, right here. Okay, Cn, H2n, On, where N is just some whole number. For the example of glucose, N would be 6. So C6H12, 2 times 6, O6. C6H12O6. And another thing to remember, guys, all sugars are carbohydrates. So another important part to just knowing things coming up on the test. And if it starts asking about sugars, you should recognize sugars are carbohydrates. And I just realized I forgot to type this in there because there's something already typed, but this right here should be a ring shape, general shape of a carbohydrate. And it's not circular, but it is a ring shape. They're all connected to each other here, forming this hexagonal ring, I guess you can say. So I didn't type that in there, but make sure that you write it in. It's a ring shape, general shape of the monomer. And here's where I said you could draw an arrow from deoxyribose or ribose. This, if you didn't know what the base was and if you didn't know what the sugar was, this nucleotide could represent DNA or RNA. The nitrogenous base is almost identical. Each amino acid has an R group that is very specific to the amino acid, but probably the most important part here is the NH2 and the COOH groups. That if you see this, COOH, carboxyl, and if you see NH2, you know this is an amino acid, you know it's part of a protein. And in general, proteins are just really long chains of amino acids. So literally, take a bunch of these guys, string the amino acids here, string them together, and crinkle them all up on top of themselves, really smash them together into some weird squiggly shape, and that's what a protein looks like. And so back to the carbohydrates, first column. Remember, if you see an ose, it's a carbohydrate like glucose. Glycogen's an example too, but not an ose. Glycogen is an example, and so is starch. We'll get to those a little bit later down. Moving over to the proteins, over here to the right. All proteins end in IN, just like the word protein. Examples that I gave you in class: actin and myosin in your muscles. All enzymes are ACEs. Again, example from the lab that we did in class is amylase. Polymers for carbohydrates are disaccharide and polysaccharide. These are both polysaccharides because they're more than one, but disaccharide, since there's a certain class of them, we refer to them as disaccharides, such as sucrose that you just saw. Polysaccharide, typically we say 3 or more, but disaccharide is a polysaccharide because it's more than one. So there's your example for disaccharide: sucrose, which is just glucose and fructose, and glycogen or starch. They're both polysaccharides, really big. For DNA and RNA, nucleic acids, that's it. That's all we're talking about. That's all that exists, at least that we know of. And then some examples for proteins: hemoglobin, we talked about in your blood carrying oxygen; amylase from the lab; and adrenaline, also talked about that one. Melatonin in your brain, making you sleepy when the lights go off. Back over here. The energy molecule for plants, we're going back to carbohydrates. The energy molecule for plants is starch. The energy molecule for animals, glycogen. Nucleic acids, what their function is, they contain all the genetic information that makes you who you are, so very important stuff. And enzymes, we're not going to talk about proteins specifically, but enzymes, we will talk specifically about enzymes and have. So enzymes speed up chemical reactions. proteins in general do thousands of things, so let's just focus on enzymes. Hint, hint, nudge, nudge for the test. Other things for carbohydrates. Sugars are hydrophilic. All carbohydrates are hydrophilic. Maybe 99.999% of them. So that means they're attracted to water. DNA just in general is a twisted ladder shape. For proteins, the R group is important because it gives the protein its final shape. These guys right here. Okay, these R groups are made of C's, H's, O's, N's. Okay, when these interact with each other, that's what causes that protein to take its final squiggled up, messy looking shape. And then also for proteins, peptide bonds. Remember, that's what's going to join amino acids together. Okay, it's a condensation reaction, but the bond is called a peptide bond. So we've only really talked about 3 bonds: hydrogen bonds and polar covalent bonds in water, in between water, and peptide bonds in proteins, between amino acids. So between these guys right here. All right, before I go into this diagram of enzyme action, go back to my, here we go, paint program. So I put in some examples here, again, something that we've kind of already talked about a few times. Sucrose, which is a G glucose and F fructose. So here's our example of what's called a substrate. Okay, the substrate is what's being acted upon by the enzyme. If you think of these enzymes as little Pac-Man creatures, then it might help to figure out what they're doing. So here's what happens. Notice the shape of these things. The shape is everything for proteins. Okay, sucrose fits right in there perfectly, and that is the substrate entering the active site. So this area right here that is perfectly set up to accept the sucrose by shape is called the active site. And then they bind to each other, and you can see this enzyme slightly changes shape, kind of wraps itself around it a little bit better. Okay, getting a good hold of it, and what you saw earlier, it's just going to add water, which is going to cause these two things now to decondense, I guess if you want to think about it that way, condensation reaction, but it's also called hydrolysis. What happens is that these guys right here, which we're sitting on here, I'm going to bring them up separately, but these guys here, that were just like this, are going to break off. Okay, this one was sitting there, this one was sitting here, but since they are now separated by this enzyme, they're going to go off and, I don't know, maybe later become sucrose, but they are no longer connected anymore. This enzyme broke them apart. It digested them, just like you saw in the lab with the starch and amylase. Okay, if this was starch, same exact thing. Okay, if this was starch right here coming in, this is what happened in the lab. The amylase went through and chewed up all the starch molecules. Okay, broke them into their separate parts so that we didn't see any more starch molecules. They were broken up, okay, into little bits and pieces. So no more starch. So let's go through here really quick and label these. That's the substrate. Again, that's the thing that the enzyme is acting upon or binding with, binding to. The active site is pretty simple, where the binding takes place. Here's the enzyme changing shape, and then the products go off, split off, and go their separate ways. And you can see by the color change, they are no longer what they were before. So that is the end result, the products leaving the active site of the enzyme. Here's an important part of this. This enzyme is not harmed. Okay, it's breaking up these molecules, but this enzyme can go back and find more. Okay, so once it does its job here, gets rid of these guys, this enzyme can go off and find some more of these guys. Okay, bind, break it up, okay, and then go find some more. So these can be reused. These enzymes are going to get reused. Right, they're just going to go over and over and over, breaking up as many of the molecules as they can find. When they run out, when they've broken them all up, then the reaction stops, because there's nothing else to break up. So that's it. I'd say if you went through this, and if you, I know it's a lot, but if you can study this, I think it's probably one of the best things to do, because it kind of sums up much of what we covered. If you have any questions, make sure you come in early or stop by during school so that we can get those answered for you and you have the best chance of being successful in this test.